Chapter 9, Part 7, Action Potential Propagation. So an action potential in a local area of the plasma membrane is indicated by the orange band in the picture here. And then there's a reversal of charge across the cell membrane. You can see that the positive on the outside and the negative on the inside. And this is kind of showing that this is a muscle cell here. And as that action potential propagates, it flips the charge, and that's what allows it to propagate, or that is the propagation of the stimulus, the action potential rather. So the action potential is a stimulus that causes another action potential to be produced in the adjacent plasma membrane. So as it goes along, it causes the next little part to be depolarized and then repolarized, and depolarized and then repolarized. The action potential propagates along the plasma membrane, showing with the orange arrow. So, all right, so I'll circle back to this a little bit. We're going to talk about it more when we get to the nervous system as well. But you have to know about how this happens in order to understand the idea behind muscle contraction, because without a stimulus, without an action potential, you're not going to have muscle contraction. So we're going to kind of revert back to the beginning, and we're going to talk about the function of the neuromuscular junction, and we're going to talk about kind of everything that happens from the beginning to the end. So again, we have synaptic vesicles in the synaptic end bulb, or the end bulb, the end of the nerve that is close to the muscle cell. We find these synaptic vesicles, and remember, within those synaptic vesicles, those little transporters are neurotransmitters. And the neurotransmitter is the substance released from the presynaptic membrane. It diffuses across that synaptic cleft and either stimulates or inhibits, depending on the neurotransmitter, because there are some neurotransmitters that stimulate and some that inhibit. The one that we're talking about is acetylcholine, will stimulate the production of an action potential in the next cell membrane, the postsynaptic membrane. And again, in this particular case, in the neuromuscular junction, we're talking about acetylcholine, abbreviated ACH. And it's important to remember that acetylcholine esterase is an enzyme that will break down acetylcholine and sends it for recycling. And that prevents an accumulation of acetylcholine in that synaptic cleft, which would be a bad thing. So acetylcholine esterase is an enzyme that is needed and used to break down acetylcholine in that synaptic cleft once it's been released. All right, so an action potential arrives at the presynaptic terminal. Okay, basically the end bulb, right? An action potential travels down, and I'm going to put this into my own words because I tend to do better that way with trying to explain things. So you have this action potential that travels along, and we're going to learn when we get to the nervous system that the same way there's depolarization of the cell membrane of the muscle, it's the same thing happening along the length of the axon on a nerve. And this is the end of a nerve here, right? This is the very tip, think of it as the fingertip of the nerve. And this is the muscle, right? The muscle cell membrane. So an action potential comes along, causes these gates, these are voltage gated calcium channels. So this action potential is going to change the voltage along the cell membrane, opening up these calcium channels. Calcium will come in and effectively force these vesicles to release their contents, acetylcholine, into the synaptic cleft. Acetylcholine will be in dire search of a receptor that matches it, and it will do a very good job of finding that receptor. When it attaches to that receptor, these gates will open, And again, an influx of sodium, which will allow that signal to propagate along the cell membrane. So keep going here. The next step is called excitation contraction coupling. And by the way, I don't know if I mentioned this because I'm kind of getting ahead of myself here. This is the first step in a muscle contraction, the events at the neuromuscular junction. So keep that in mind. The next step in a muscle contraction is called excitation contraction coupling. I do think there was a Big Bang Theory, the TV show Big Bang Theory. I think there was an episode called excitation contraction coupling. So this step links the electrical and mechanical components of contraction. So basically, it turns the electrical stimulus into a mechanical movement. So the action potential produced on the sarcolemma, and I'm going to go back to this one. So this would be the sarcolemma of the muscle. So that action potential is going to follow that sarcolemma. And if you remember, when I talked about the T-tubules, I said the T-tubules are an extension of that sarcolemma. So an action potential produced on the sarcolemma will go down into the T-tubules, and the calcium it'll force the calcium channels on the sarcoplasmic reticulum to open. Calcium will enter the sarcoplasm, which is the cytoplasm of the muscle cell, and calcium, which has a strong attraction to troponin, the calcium will bind to that troponin and result in a muscle contraction. And we're going to get to that here. So an action potential is propagated along the sarcolemma and down into the T-tubules. That allows calcium to be released into the sarcoplasm. Calcium ions, which love troponin, will bind to troponin. And when something binds or unbinds to a protein, it causes it to change shape. So the troponin that is, remember, it is attached to or bound to the actin molecule, the troponin will move. And troponin is also attached to tropomyosin, and that causes it to move as well. So not only will troponin move, but tropomyosin will move. And if you remember, the tropomyosin was blocking the sites on actin that myosin needs to attach to. So when the calcium ions bind to troponin, troponin will move, tropomyosin will move, and now those active sites will be revealed and open. Once those active sites on actin are exposed, the myosin filaments are able to bind to them to form a cross bridge. And of course, ATP is needed for this to happen. But either way, they're revealed, ready to go, able to form a cross bridge, which is the last step of muscle contraction. So during the cross bridge cycle or movement, we have exposure of the active sites before the cross bridge, calcium binds to troponin, and the tropomyosin move. Active sites are now revealed. Cross-bridge formation is when myosin heads bind to the exposed actin sites and form a cross bridge. So remember, we needed ATP for this to happen. And as soon as ATP attaches to that myosin head, it breaks it down into ADP and phosphate, because adenosine triphosphate means there's 3 phosphates. Adenosine diphosphate is 2 phosphates because one was removed when it was broken down. where am I here? So cross-bridge formation, the myosin heads bind to the exposed active sites on the actin filament to form cross bridges, and then the phosphates are released from the myosin heads. And that, when anything binds or unbinds to a protein, it causes it to change shape. So when that phosphate is released, the power stroke happens. Energy stored in the myosin head is used to move the myosin heads, causing the actin myofilaments to slide past the myosin. So look at this picture, because I know my words are probably confusing. So here we have the active sites available. We have ATP that comes in, which is already broken down at this point, and they can attach, right? So now they're attached. When phosphate pops off, this lurches the head of the myosin forward, pulling actin towards the center of the sarcomere. Remember that picture when I went back and forth, showing that the actin moved from here to here? Well, that's happening because these guys are pulling it, right? The little myosin heads are pulling the actin towards the center of the sarcomere. Z disc to Z disc. So I think of this as the myosin heads are kind of pulling hand over hand over hand over hand, pulling bloop, 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 the actin towards the center of the sarcomere. So, and this is describing how that's happening, how ATP is needed, and ATP is broken down into ADP and phosphate, and how phosphate and ADP are released and what that causes. So, as I mentioned, when the ADP molecules are released from the myosin head, then the cross bridge, or then we can have more ATP come in, and when that happens, the cross bridge formation is broken. So the ATP molecule binds to each of the myosin heads, causing them to detach from the actin. So I'll take a little sidebar here. So without ATP, cross-bridge release cannot happen. Detachment cannot happen. So when someone dies, you may wonder, why does their body get stiff for a period of time? There's a certain amount of time that it's not, and then there's a period of time where the body becomes very rigid. And it's referred to as rigor mortis. And rigor mortis happens because there's no ATP to break those cross bridges. So without ATP, that's what causes rigor mortis. All right, so here we are. We're kind of, it's back into its cocked position, ready to go, because ATP is attached. It is broken down into ADP and phosphate. They refer to that as the hydrolysis of ATP, and that allows for cross-bridge attachment, and then the whole cycle goes over and over and over again. A couple of the videos that I posted are going to be helpful with this, and I will talk about this again. But for now, we will pick up on the next installment of this chapter.